Hello everyone. Today I will be presenting our new journal club paper titled as Mitochondria serve as holdout compartment for aggregation-prone proteins, hindering efficient degradation, published by Gerish et al. And it's a very recent publication. It focuses on the inhibition of EIF5A, which we will learn about, and its translational insights into being used in neurodegenerative diseases, which are sometimes caused by aggregation-prone proteins, such as Huntington's disease and Parkinson's disease. So, the mitochondrial holdout leads to the accumulation of these disease-related proteins, and the authors are trying to find a way to tackle with it. So, we can get started. Okay. So, as you all have read the article, or as I'm hoping, the authors choose a reporter system that combines a strong promoter, a stable reference protein, and an aggregation-prone protein. Now, when this when this vector goes through translation, the P2A motif in between results in the separation of the two proteins, the stable and the unstable one, and in result you have two proteins per translation of each vector. So that way we know that for one stable protein, another unstable protein is produced, and by comparing the ratio of blue fluorescent protein generated by the stable protein and the yellow fluorescent protein ratio, we can determine how much each protein is retained in a cell, accumulated, or degraded. And an important thing to know about the YFP CL1 reporter is that the CL1 includes a helix that is amphipathic, means that one part is hydrophobic and one part is hydrophilic. And this is a common characteristic of aggregation-prone proteins, which make them bindable to the mitochondria. So let's keep that in mind. And also natural mitochondrial targeting signals are also amphipathic, like the structure of these reporters. So we know that this reporter can localize to the mitochondria, can act like our aggregation-prone proteins, so we use them. And now we want to check what happens if they accumulate in the cell properly. So in panel B, you can see that two groups are treated with epoxomycin and also a drug named cyclohexamide. The epoxomycin is a proteasome inhibitor, and we'll be hearing about the proteasome, especially the ubiquitin proteasome system in this paper a lot. To give a brief introduction, the ubiquitin proteasome system degrades the cell's unwanted proteins. It's not only that, the proteasome has many vast applications and functions, but it's important to remember that it acts as a protein degradation mechanism, and it's very important. If it gets disrupted, then proteins start to accumulate and form aggregates. So, which is why researchers are trying to find the links between the proteasome, the ubiquitination system, or some defects in it to see how these lead to neurodegenerative diseases. But until now, drugs usually affect the proteasome in that they inhibit this mechanism. So it's actually harder to promote the proteasome to work more, do more protein clearing. And the authors suggest that maybe the proteasome in a spatial level isn't doing its max proteasome clearing capacity, maybe because there aren't enough substrates or it can't be activated as easily as one may think. So, okay, and epoxomycin is a proteasome inhibitor. So in panel B, we can actually see that with time, when the proteasome is inhibited, the unstable protein, our YFP-CL1, accumulates in the cell, while our stable protein, similar to actin, which is a reference, is in the same level. And once cyclohexamide, which stops new protein synthesis, is administered to the cells, after a while, because no new protein is being translated, the already existing unstable YFP-CL1 proteins degrade, and there isn't much left compared to the epoxomycin group. And once again, our stable protein levels and actin levels are unchanged. Okay, the Researchers also validate this by doing some flow cytometry, and they see that by looking at the x-axis, yes, the blue fluorescent protein levels are the same, but the difference is being caused by the yellow fluorescent protein levels, with the highest levels in the epoxomycin group and the lower levels as expected in the cycloheximide level group. Okay, and also from the supplementary, I wanted to include an image to see in the epoxomycin group how the YFP-CL1 proteins actually form aggregates. So these aren't soluble or in the liquid form proteins. These have actually produced solid aggregates, very similar to what happens in huntingtin, for example. Okay, so now that we have established our reporter system, we can move on to, I think, a very interesting method that the authors did. I was very excited to learn about this. What they did is basically a genome-wide CRISPR screen, and they have siRNAs that target every single gene in the human genome. So they have a Cas9 reporter cell line, and by this method, they can knock out for each cell, only one human gene is knocked out. I think it's mind blowing, but and then they wait for 10 to 18 days for these genes to be transcribed and translated, and then they do fluorescence assisted cell sorting to have two distinct populations, one having YFP-CL1 high levels and the other population having lower levels, because they want to see which cell genes, when knocked out, lead to the accumulation of YFP-CL1 protein, and which genes, when knocked out, lead to lower levels. So what does this mean? In the YFP high population, the genes which have been knocked out stop degradation, and had they been there, the YFP-CL1 wouldn't have accumulated. Also, like similarly or vice versa, in the YFP low population, the genes which have been knocked out accelerate degradation. The knockout of those genes accelerates degradation. And this is also confirmed by the relative fluorescence levels. Yes, the YFP-CL1 high group has higher YFP levels, and the low group as well. So after the collection of the two distinct populations, sequencing is done, and then by data analysis, they identify some gene targets to see which genes may be useful in their search for these aggregate for therapeutics for these aggregation-prone proteins. Okay. By the way, they performed this screen 2 times, and what interested me was that in the YFP-CL1 high group, they want to show that the two replicate groups overlap at 824 genes. But I think it's a bit interesting because this is the same experiment done twice, but I think the numbers are a bit too far off. Like, yes, the two groups have 824 unique hits, but also they have nearly in total 700 different hits. So I would have expected to be more of an overlap. I think this is definitely worth discussing. Anyways, first they analyzed the YFP-CL1 high group, and by the gene ontology analysis, they see that the most common, I can say, or the categories that are most seen are about membrane protein insertion to the ER, or some processes related to protein ubiquitination and degradation, which is okay, maybe interesting, but maybe not that surprising, because what happens is that when they look at the YFP-CL1 low groups gene ontology results, they see a lot of processes related to mitochondrial organization, function, health. So this is unexpected because okay, like proteins get ubiquitinated in the ER and then they're degraded by the proteasome. But what is the role of mitochondria in all this? So they move forward to investigate this further, and they identify possible gene candidates to study by doing the log fold change analysis. And in the right quadrant, you can see the mitochondrial genes and the selected genes. I would only just say that only two actually of the genes aren't labeled, but I think it would be nice to label them and provide why they didn't move forward with it. And as you can see, um, this is the most simple way I can put it, very far away from them, which is obviously means something that EIF5A, a gene, is located. So it means that its log fold change is very different. So the researchers will focus on that as well. To further investigate this, the researchers knock out the selected genes one by one in human cells, and then they proceed with a different approach. They use four different fluorescent reporters, and they look at the change at the levels of these different reporters. They check to see some ER reporters are obviously trusted YFPCL1 reporter, and others related to cytosolic reporters degraded by the UPS system, and other pathways. And then they group the reporter level changes group by group, and they see that the EIF5A group stands out because the yellow means that that reporter's levels have dropped. The black means that the levels haven't changed, and the blue actually means that it has accumulated. In EIF5A's group, they see that the YFPCL1 reporter level has decreased, while the UBG76V group has actually increased. So there's a dual role of EIF5A. So there's like a double control, an opposing role. So they investigate this further, focusing on EIF5A as the target. So here, what they do is they want to see how EIF5A regulates the protein degradation. So first, what they do is they knock out it from the cells as the gene, and they see that yes, there's a decrease in the YFPCL1 accumulation. And they also use a drug named GC7, which inhibits the EIF5A activity, and they see that similar to the genetic knockout, um, a chemically induced treatment also affects and decreases YFPCL1 levels. So the researchers ask if the chemical and biological genetic pathways work together. Do they target the same path, or are they acting together? And in panel E, we can see that together they don't have an accumulated effect. So yes, both genetically and chemically, these should be GC7, and the knockouts should be affecting EIF5A inhibition in the same way. Also, I added a figure from the supplementary in the bottom, as you can see. Hyposine is an amino acid that activates EIF5A, and it's a very rare amino acid, like a process, and it's only found in the activation of EIF5A 1 or 2, as far as I know. So they look at it, and in the left panel, you can see that the genetic knockout, and in the right panel, the administration of GC7, both lead to the decrease of hyposine levels, and obviously done by inhibition of EIF5A. EIF5A's levels decrease, while our references are stable and remain the same. Okay, so now they move forward with how YFPCL1 is cleared. So they use the control group, the epoxamycin tag 243, which is a ubiquitination inhibitor, and BAFA1, which is an autophagy inhibitor. And here they see that yes, in the control group where the EIA5A is knocked out, the definitely the YFP levels have dropped, However, when epoxomycin is administered and the ubiquitination inhibitor is administered in the third graph, these levels are rescued. So this means that the protein clearance does heavily depend on ubiquitination and the proteasome, so the UPS pathway, but they don't see a significant change in the uh, autosomal or lysosomal inhibition part in the fourth graph. So they conclude that maybe it doesn't have much relevance. And then they move on to see if the location of the proteins matter. So they use cytosolic reporter substrates in part C and nuclear reporter substrates in panel D. And they see that while there isn't much change in the nuclear, the proteins sequestered in the nucleus, there is a significant decrease in the cytosolic proteins, which does make sense because these proteins, these cytosolic proteins, are more likely to be ubiquitinated in the ER or held by the mitochondria. So once EIFA5A is inhibited, they would be released to the cytosol, ubiquitinated by ER, and then degraded accordingly. So actually, localization does matter. And by the cytosolic localization, the degradation does increase when treated by GC7. And they also check the proteasome activity when GC7 is administered and when EIA5A is knocked down, because they want to see if there's an increase, decrease, which would mean that maybe EIF5A isn't as effective, but there are no changes in the proteasome activity, which means that it is normal level. There is no increased activity or decreased activity, like the capacity isn't increasing, just because the available substrates, the aggregation-prone proteins released from the mitochondria are increasing. They're also being degraded normally, like not by an increase in proteasomal activity. So, the genetic or chemical inhibitors of EIF5A does increase the ubiquitin-dependent proteasomal degradation of YFP-CL1, but not by activating the proteasome. This is a totally different pathway, we can say. So, our authors are wondering how does this work? And first, they want to make sure that YFP-CL1 can localize to mitochondria. And by verified in panel A by the mitochondria diatim 50, they can see that there's co-localization to mitochondria. And usually in normal conditions, these degradation bound proteins should have been first ubiquitinated at the ER and then sent to the proteasome. And by fractionation, they find that yes, these proteins can be found in both pure mitochondria and ER. However, this is confirmed by the Western blot at panel B. But in panel C, we can see that unmodified proteins reside in pure mitochondria, while in the ER we see a laddered appearance, like a laddered band, which is similar to the pattern ubiquitinated proteins have on the gel when they walk on the gel. So this means that if the proteins can be held on the mitochondria, they have escaped ubiquitination by the ER, and which poses a problem because these proteins won't be destined to the proteasome for degradation. So I think this is very important because they escape ubiquitination, they're held in the mitochondria, and by accumulating there, they lead to aggregates or other problems disrupting cellular activity. However, we see that when EIF5A is knocked down, this results in a mitochondrial network change. In this remodeling in panel D in the left bottom, we can see that the normal mitochondria is closer, but with higher levels of GC7, which inhibits EIF5A, is administered, the mitochondria becomes more fragmented. So what happens is this: when EIF5A is inhibited, the mitochondrial network is remodeled, and then by becoming more fragmented, the proteins that were held captive in it, that were hiding out in the mitochondria, can be released to the cytosol, and then the degradation accessible pool in the cytosol increases. Thus, the YFPCL1 levels decrease as these released proteins can be ubiquitinated and degraded by the proteasome. So the mitochondria does not no longer hide them. Um, Last but definitely not least, our authors want to confirm that this is the case also for disease-associated proteins such as mutant huntingtin and α-synuclein, which respectively cause Huntington's disease and Parkinson's disease. And what they do is they first try this with the drug they had used in YFPCL1 protein, GC7, and they administer in cells containing soluble mutant huntingtin and also with cells containing mutant huntingtin aggregates, which is the case in Huntington's disease. It forms aggregates, and they see that the levels do increase in when EIF5A is inhibited. So this is definitely good news. Both the soluble and the insoluble fragment decreases. So this means that this pathway, this inhibition by this chemical inhibition can be used in the treatment of Huntington. And then the researchers asked the question if it can also be done genetically as it was done for YFPCL1. And they used EIF5A knockouts and see that yes, by as you can see in panel D, the mutant huntingtin that is causing the fluorescence levels does decrease, and upon the administration of epoxomycin, which inhibits the proteasome, these levels can be slightly increased, so they can be recovered. So this is great news because they have found a way to degrade the proteins that had been accumulating and causing real life diseases. They also tried this for α-synuclein, which also has an amphipathic helix, just like mutant huntingtin, and leads to Parkinson's disease. And in EIA5A knocked out cells, they also see a decrease in GFP levels caused by the aggregation of α-synuclein. So they have seen that both genetically and chemically, these EIA5A can be inhibited to prevent real life neurodegenerative diseases. And I think panel G provides us very solid um, very solid evidence for the use of EIA5A knocked out cells because the portion of the mutant huntingtin GFP protein that had been localized to the mitochondria has decreased 42%, which I think is high. Now 42% of those previously held out hidden proteins are now in the cytoplasm, available for the proteasome to degrade them. And I think that makes a big difference. And authors have also used more therapeutic drugs which inhibit hyposination. As we talked, this also inhibits in turn EIA5A, and it does decrease the aggregation-prone protein levels. So these findings can be applied to real life and they can be used to find some therapies for neurodegenerative diseases such as Huntington's and Parkinson's. So I think in this paper, the authors set out in a different way, but then changed their direction towards mitochondrial implications of EIA5A in protein degradation, and they connect mitochondrial function, health, and networks to protein degradation and the ubiquitin proteasome system. I tried to tell you about it and I tried to explain it, but please feel free to ask any question, any questions, any part of the article that you couldn't quite get. I will try my best to explain it because I also learned it along the way. Please tell me anywhere that is not understandable, and give me some feedback. I'll be looking very forward to hearing from you. Thank you very much for listening and see you in our journal club discussion.